All right, welcome everyone to this webinar today. My name is Maria. I'll be facilitating the webinar. We're going to be talking about microbiology and teaching microbiology with Labster, of course. My guest today is Ana Barral. Welcome, Ana. Just briefly muted for a second there. Ana will introduce herself in a few seconds and tell you why she's here. She is using Labster today, of course, so that's why we brought her in. Just a few practical details before, and that is one, this webinar will be recorded. So of course, we hope that you stay with us for the whole time, but in case you do want to watch it later or you want to share it with peers or you have to leave early or something, I will be sending out the recording tomorrow. So that's one thing. And I will be taking questions throughout the whole webinar as well. Feel free to type them in the question box or to put any comments in the chat section, and I'll try to have a look at them while we go through the webinar. We will also have time after the webinar for a few questions in case you want to wait till the end. Let's see what else. Yeah, one more thing as well. We are going to focus more on sort of teaching with microbiology today rather than talking about the specific simulations, because some of you might know that we do have a new microbiology package with a bunch of new simulations in there, and we did present those a few weeks ago in our What's New from Labster webinar. If you want to check that out, you can go to www.labster.com/webinars. There is also a handout, you might be able to see it in the toolbox there, that has all the new simulations with a little description and a few screenshots in there if you're interested in learning more about those. But Ana has been teaching with some of our sort of older simulations, and so we're mostly going to be talking about sort of, in general about teaching with those simulations. So let's get started. Ana, tell us a little bit about yourself.
Hi, everybody. It's good morning for me from California. So I am a professor of biology at a private nonprofit university called National University. And I teach all kinds of biology, so general biology, both for majors and non-majors, anatomy, physiology, microbiology. And who my students are, they tend to be, well, they are undergraduate students, and they usually tend to go to health careers. So I have lots of pre-nursing students, but also other disciplines. Now, in the slide, you can see that I have used Labster for introductory microbiology and also Survey to Biosciences. This is like a non-majors biology class, and the number of students may seem a little bit too low. So my class was very small, but as I will explain later, this was part of a study when several instructors in several sections were testing Labster.
That's great. Thank you so much. And again, if you guys have any questions for Ana along the way, please feel free to just type them in the questions box, and we'll fill you in. So first question here is, and I'm just going to briefly tell you what you see here is a screenshot from one of the new simulations, and I'll be showing some of those along the way just to sort of brighten our slides up a little bit to make them look a little bit more interesting. But yeah, let's see. I don't think people can see the question here, but it says, how did you get started with Labster? Ana, could you tell us a little bit about that process?
Sure. So as I was telling you, we have, I teach all kinds of biology, and particularly microbiology, for a number of undergraduate students going to a number of different health careers. And one of them is public health. And the public health program people wanted to be able to offer the whole program online, fully online. So that required us to figure out how to teach a microbiology lab course online. Also, in my university, there is an organization or a group called the Precision Institute, who are in charge of finding technological innovations to enhance student learning, the use of data analytics, and so on. So they helped us to, look for different offers and possibilities for these online labs. And then we had a demonstration by Labster. Now, I also want to say that we are not new to virtual labs. We have tried several companies and also resources available online, and we were very impressed from the beginning with Labster. One of the reasons is because they had actual data of effectiveness. So there are a number of articles, and I think Maria has posted them somewhere, that show that those in several courses where students used Labster, the student learning was better. So we decided to give it a try. We tried different simulations and then chose a number of them, and I would tell them later about them, but it's basically very kind of common microbiology labs: quantification, identification, also sterilization, fermentation lab, and a lab that was dedicated to like sterilization, pasteurization, and so on.
Yeah. And like you said, we do have a bunch of research papers that are published about Labster, and they're actually on www.labster.com/research if you want to check them out there. The one that you referred to here is in your testimonial, which is on our blog, but we'll also send that out in the email tomorrow with the recording in case you're interested in reading that. I just also want to add here that I think it's quite common actually for these, pilot, for a lot of the professors who do start using Labster, or the schools that do start using Labster, to do it through a pilot study like this. And it's really great, and this is a piece of advice I think we should share with everyone, is that, it's really great to document the effects of Labster when you do start, and really look at, where the students are when you start and where they are after. I know not everyone does that, but it does help schools after that pilot study to continue using the virtual labs, because you do need to find the funding for it, and the best way to do that is really be able to document the effects of it, right? So, but yeah, we'll get back to that. Yeah, sorry.
Yeah, if I just adding to what Maria was saying, I come from a research scientist background, working in the lab, test tubes, animals, cell lines. So when we become more like education oriented, we can keep doing the same thing, research. You want to try that something is better than the other or the same way, you should use a scientific approach and, testing, control, and experiment. So we are going to show some of these results later, but indeed the best way to make your case for using a certain approach, a certain tool, is to actually prove it with data that yes, it's more effective, the students like it, the students are learning more, and so on.
Yeah, and we do have, because there are so many studies by now that actually are using Labster, it's easy to describe some of the questions from there and really just make the job easier by replicating those. So, but yeah, let's move on to the next question here, which is, again, I'm sorry if you can't see if it's cut off at the bottom there, but how do you use Labster in your course? Tell us a little bit about that.
Yeah, so this was a pilot, so we haven't done the fully online lab yet. And in the pilot, what we decided is to use the simulation as a pre-lab. So we kind of match the virtual labs to labs that we would be doing in the lab, and then give them as an assignment, as a homework for the students to do. One thing, so this was the older Labster labs, and so they wouldn't be applied in the lab until a few weeks into the course. So we introduced them early on to the students so they could get more comfortable with the interface and just using the controls and so on. So again, this was like a homework. Students were working from home, trying the simulations. But also, once they completed the simulation, we really wanted to bring them back to the discussion. So for example, if they did it as a pre-lab of a real lab, once they got to the real lab, we would discuss, like, do you remember when you did this and this, and the results you had in Labster? Well, this is exactly what is happening now. So, make the real life connections to see how relevant it was for the lab activity. And then there was one lab that we tried that has no real life equivalent, which was a BSL-4 lab. Like, how would you handle Ebola or some infectious agent that is very dangerous? So in that case, we will refer to it in the lecture. So do you remember when you did this and this and this in the lab? This is because this agent is so dangerous, infectious, and so on. So it's a nice way to show lab activities that are not available in most schools, I would say. I don't think many schools have the ability to deal with BSL-4
Yeah.
students.
Yeah, and I remember you told me that when we spoke the first time that you'd also entered one of these labs in a virtual situation for the first time, and it's quite an experience. And I think that's something that's really cool that a lot of students appreciate is the chance to get to try some of these things that are usually too dangerous or too expensive to try out in real life. We do actually have a question here already for you, which is, have you tried labs for upper division MICR labs for majors, for example, medical microbiology, and what are your experiences there? So I don't know if you have.
I have not,
No.
sorry. I'm trying to think. We have been discussing, and again, as I said, we started with these courses and these labs because there was a direct request to use it for these undergraduate courses. But I think that it's probably much easier to use in upper division courses because the labs are pretty sophisticated. You know, again, coming from a research background, I can appreciate that how real life they are, you know, when you enter that environment. So I think there is no major issue for upper division courses, but I haven't done this personally.
Thanks for answering that question. And we, in the meantime, also got one more, which is, do you plan on using Labster in, I can't see what it says there, of hands-on lab or only as a pre-lab? So I guess in replacement of the hands-on lab or in supplement to.
Yeah, so the plan is to develop a microbiology lab course which is fully online. And again, as I said, this would be for public health students. So we are going to keep the Labster as a pre-lab for hands-on labs for, let's say, nursing students or for majors. But for public health students, which, you know, they don't really require that much hands-on experience and the program wanted it, we will develop a fully online lab course when there will be, you know, a much closer association between the material taught online in the lecture and the labs that they will be doing online.
Yeah, cool. Thank you. All right, moving on then to, let's see. Sorry, I have a lot of windows open here to administrate questions and stuff. So if I'm looking around the screen, that's why. But let's move on to this next one, which is, I'm sorry, I can't see. How did you, I'm just waiting for this. There we go. How did you make your class engage with Labster?
Okay, so here come the practical details. You know, everything is about the details. So one thing is that as instructors, we should do the simulations ourselves. That's really important, not only because it makes it easier to help students if they have any issues, but also to understand how it works and again appreciate it. When I did the BSL-4 lab, I was very impressed in the level of detail of handling Ebola. I think it was Ebola or anthrax, one of those evil ones. So instructors, do your homework before giving it to the students. Second is that start with an easy one. So in our case, there is this pasteurization, sterilization lab. It's kind of it starts with a fun story of a farmer who has to pasteurize her milk and it has some issues. So it's very simple and it, you know, many of the concepts there are common sense concepts, so it was not extremely demanding. So you kind of look for the simplest one, the easiest one, and then you assign that first to the student. Third, it helps to demo it in the classroom. So I brought it up on the projector and I clicked through and I showed them some of the controls and how, you know, where to find the information they needed. So if you haven't done Labster, you follow steps, but if you get stuck, you don't know what to do, there is a background information that then you can read. And most students want just to do it. And then you have to say, hey, if you cannot do this, you want to read information first. And it's very, you know, similar to whatever we'll be teaching in a lecture lab. and allow plenty of time. We have seen in our courses that most of my students are older students. So young students are usually very comfortable with games, online games. So usually they find Labster easy to navigate and they like it and they think it's very fun. But you may have some students who, for X number of reasons, they are not familiar for them, they have technological issues, etc. So just allow time and check in with the students before assigning it as a required assignment. So be sure that everybody is more or less comfortable, their issues have been solved, and then go in to, as an assignment.
Yeah, that was a lot of great advice. I think that's really useful for a lot of people who haven't used virtual labs before as well in general, not just Labster, but really making people comfortable with an EdTech tool. First of all, becoming comfortable with it yourself so that you feel confident when you go and use it with your students, but then also making sure that the students are comfortable using it and really, getting them engaged with it. So that's really great advice. All right, then let's move on to the next question here this is an interesting one. So pros and cons of virtual labs, what would you say there?
Well, I would say among scientists, there is this overarching skepticism about virtual labs, like how can you, replicate what is going to happen in a real life? And there's part of it, there are things that cannot really replicate in a lab, and a lot of it has to do with errors and troubleshooting. And actually, that was one of the things that we liked about Labster. It's not like click, 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 and everything is fine. So if you know microbiology, you know that when you spread bacteria on a plate, you have to try different dilutions because some plates may get empty and others completely full. You need to try different dilutions to get the right numbers. So that is shown in the simulation. So it's not like students spread the plate and everything is pretty. They have to decide which of the different options is good. So that is obviously a con that is not going to replicate 100% what happens in the real lab. On the other hand, one pro, and for me that kind of is important, it's virtual labs are democratic in the sense that it would allow students who don't have the opportunity to, access laboratories or, research labs, or schools that don't have the resources to, compete with laboratories or schools with more money. Actually, they can provide a similar experience. And again, going back to BSF or lab, I have never been in a BSF or lab. I have been in labs where I had to take all kinds of cares and precautions, but never at the BSF or level. So even for me, that was an equalizer, like, oh, this is how it works. And needless to say, for schools that have, financial issues, it may solve the problem of teaching laboratory skills to students, even if you don't have a lab or if you don't have enough equipment. So I think when considering the pros and cons, there are valid, good reasons to use virtual labs.
And when we spoke as well, and I got a question that I'm going to get back to in 1 second, but when we spoke as well, you said that there's, virtual labs in general have pros and cons, and there's Labster that has pros and cons. And maybe you could mention a few of those in a second. We got a question here if we could see a demo. So I really want to show one of the simulations that you have been using, which is bacterial quantification by culture. So this is just a quick sort of a screencast, showing it's a video recording basically of the simulation. So it's not, of course, how the whole simulation goes. We're going to go back and forth and see a few things in there. But it's a short video, and I'm going to show it here live. And we do sometimes have a few issues with people not being able to view them because of Wi-Fi or other reasons. So I'm going to copy the link into the chat box as well, in case you aren't able to view it. Here we go then. the bacterial quantification by culture simulation, your students get to experience what it's like to work in a real lab. First, they're sort of prepped for the lab. They have to put on a lab coat, and once they're ready, they get to enter the lab and start experimenting. They're given a mission, which is that their virtual colleague has just returned from an expedition to the Amazon rainforest and has brought back his promising sample, which is a fungus that seems to inhibit bacterial growth. So the student's mission in the simulation is to quantify the effect of this antibiotic compound on bacterial growth. But they'll only be able to succeed their experiments if they manage to combine smart experimental design, some simple mathematics, and proper microbial culture techniques. So the students are quizzed along the way and can review theory on the virtual lab pad that you see here to help them answer the questions. And fast forward a little bit, and then the students are let off into the lab to experiment. Their task is to use the available equipment to test if the fungal compound has antibiotic properties. So they have to first prepare an experiment, and you can see the students here get to try out the pipette to inoculate tubes with E. coli cells. So they'll have to figure out how to quantify the bacterial cells by inquiry-based learning. And the idea is that they'll learn much more by trial and error. And if they're fast learners, they'll be challenged, or you might even say rewarded, with some extra questions. The next step of the simulation, the students will incubate the bacteria so that they can observe their effect on the fungal compound. So you see the incubator over here. Normally, you'd have to wait 16 hours for the incubation, but because we're in a virtual lab, we can speed up the entire process and skip the waiting. And the students can then observe the tubes and are prompted with quiz questions to test their knowledge. And next, the students will get the task of quantifying the bacteria in order to determine if the compound is any good as an antibiotic. So fast forward a little bit more. And they can count the number of colonies in their samples, and from that extrapolate how many viable cells there are with a virtual calculator. The students are also given the opportunity to perform a serial dilution. At the end, the students will then know how to calculate the colony forming units per millimeter and of the bacterial culture by using a CFU calculator. And they'll be able to see from all their experiments and calculations whether or not the fungal compound then can be used as an antibiotic. So there's a lot of experimentation and calculation that goes on throughout the simulation, as you see here. At the end of the simulation, it ends with their findings being sent off to a pharmaceutical department for further investigation. And the student is informed that they might just have built the foundation for an antibiotic that could save millions of lives. So this really establishes the importance of what they've just learned and connects it to the real world. And that's pretty much it. All right, I hope you were able to view that video. And that what I said wasn't complete nonsense. I'm not a microbiologist, just so you know. So I hope I explained the simulation well enough. I just want to say that it is possible to see all our simulations, like demos, on the faculty resources page. I think everyone who has an access to Labster has an access to the faculty resources page as well. And you can also get like a demo of that in case you're interested in going in and seeing all these screencasts. I got a good question here while the video was showing, which was why are gloves not worn? And I actually don't know if it was necessary. Usually it is like a requirement. You know, we do try to make sure that the students know when to wear gloves and when not to wear gloves in the simulation. I don't know, and this is also a screencast that was, we do a lot of the screencasts in alpha versions instead of the final versions that are released. So maybe that's why. I don't know, Anna, do you have a good response?
I don't remember if that was, I don't remember it on the gloves. I mean, I do recall in the BSL-4 one that I've been referring to that there were like 2 gloves. You had to, they went through the whole thing of double gloves and then how you had to discard between steps. So there may have been a glove, it just didn't show. That little screencast showed what I was referring before about the number of colonies. You could see how some plates were completely full and others were, you know, the right or an appropriate number of colonies. And Maria was referring to pros and cons in Labster specifically. So one of the pros of Labster, and I have seen this through a number of simulations, is that you get numbers with it. And again, we have tried many different simulations in the online and through different companies, and it's really nice when you can get data, because especially undergraduates, they struggle with math, like, you know, calculating exponentials, CFUs, like it was shown there. It's very difficult for them. So the fact that besides, you know, doing the process of, let's say, bacterial isolation, etc., you also have to calculate numbers, and some simulations require you to do graphing. So those are pros of Labster that I appreciate, because I know how much students struggle with math and calculations.
Yeah, yeah, and that was, I guess that's also one of the things that was cool about this simulation, right, was that little virtual calculator. I do know that some of our simulations and some of the new ones, I think, as well, even have 3D versions of the data. So you, like, some of the simulations require you to plot data and then visualize it into this whole 3D graph, which is pretty cool as well. I'm not sure if it's the microbiology ones, but something to look out for at least. I'm getting a lot of questions here, and I just want to actually save them till we, unless you have any more pros and cons that you want to mention here, Anna.
Well, I think the numbers I was just going to add, so this is about microbiology, but I have used some simulation for the non-majors general bio. One of my favorite ones was about lactate, lactic acid in exercise, and you know, it involved both soccer players and rats, but you got numbers, and it was really nice that you would measure oxygen and lactic acid concentrations in real life graphs. So depending on the simulation, as Maria was saying, some of the graphic data are very advanced and very, you know, real life like.
Yeah, realistic, which is nice. It's also nice for students. At least I remember when I was a student, you know, being able to, well, that question you always ask is, how can I use this in the real world? And if you get the sense that it's connected to the real world and get some context on there, at least I really enjoy these, like you said, this in the bacterial, was it bacterial? No, the one with the chicken farm.
Pasteurization.
Pasteurization, that's the one. Yeah, having this sort of context applied to it, I think is really, really cool. Anyway, so yeah, let's move on then here to the last one, which is what outcomes have you seen? And this is what we touched upon a few minutes ago with the research that you've done. So maybe you could tell us a little bit more about that.
Yeah, so we piloted the Labster as a pre-lab activity in a number of sections. I forgot how many, but it was a total of 60-something, 69 students through different sections, different instructors who use Labster, and then control sections. So these were just business as usual microbiology sections. And then we used a quiz that was actually published in one of these articles that we mentioned before that looked at the effectiveness of Labster. So we used a quiz that addressed a number of issues related to that laboratory activity, and that was bacterial isolation. So this is a lab that is done in the lab. So the control sections did it in the lab, and the Labster section did it in Labster and in the lab. And we saw a significantly higher score for those students who, you know, completed it in Labster. And some of the questions were easier, but some required critical thinking. You know, it was like, if it was positive in this test and negative in this test, what do you think it is? So it was an application question. So that was good. You know, we want to run some more tests and, you know, confirm it, but it was statistically significant. So that was good. We also ask, you know, both students and instructors about their experience. And as I mentioned before, so we have students who are very comfortable, who loved it. It's like a game, so much fun. And then some students who complained, like, well, why do I have to do that? The internet, the connection, my computer, etc. So just be aware that in order to run the simulations, there has to be a reliable, strong Wi-Fi or internet connections, and the students shouldn't be, doing Facebook and other things at the same time because you require bandwidth to run them. But basically, overall, it was a positive response for students and overall a positive response from instructors. So again, some instructors felt it was new and why do I have to do that? But some of them were very positive and said, oh, I want to explore in other disciplines too.
That's great feedback. I think, yeah, definitely, technology and internet connection is always something that's going to get in the way of some user experiences, and that is a shame. And I think with time, hopefully, stronger and stronger internet and better technology is something that's going to get us to a place where that won't be in the way of a good experience. But that is really great to hear that most of the people at least enjoyed it still. And I also think, just getting used to the technology, like you say, not having other windows open and these different small things that you can do to make sure that it's working as best as possible is always good. Good advice as well for other peers. So yeah, is there anything more you want to add to that study? You said you're going to keep on doing some research now, and or
yeah. So we were talking about data analytics before, so there is a way Labster can do that they can look at students' activity when performing those simulation labs. So there may be students who go in and breeze through it in 15 minutes, and others may be struggling and going back and forth. So some may start and then go and have dinner and then come back. So we want to have a little bit better insight of how they are doing the simulations and which are the difficult spots, because it will help us also. Let's say we run this as a pre-lab activity and we see, oh, everybody got stuck in this point, let's say calculating the CFU, so we can go back and do some extra work, little discussions about and practice how to make those calculations. So it's going to give us a more granular view of what are the problems, what are the concepts that students have difficulty with. And we are also opening to try in different, like we are looking now to physiology. So we want to incorporate some physiology labs. And in physiology, we have less laboratory resources that we have in microbiology. So I'm pretty sure that we can bring in some more advanced physiology labs to enrich the lab experience besides whatever small things we can do in the lab. So yeah, we are opening and
opening up,
looking at more labs.
Super interesting. And I just want to those of you who are new to Labster, I just want to say that, yeah, what you're talking about here, viewing sort of the performance and seeing where students get stuck is something you can view from a Teach dashboard. Of course, if you have LMS integration, you can view these things through there quite easily as well. We do have like a pretty updated Teacher dashboard. I don't know if you use that, Anna, but we do have one that is being updated all the time so that, we give teachers insight into all these different stats and being able to see how their students are performing, how long they're taking, and where they get stuck and so on. So it's pretty cool.
I just remember now you said the dashboard. So when we started using Labster, we were worried that students would get stuck, couldn't manage to do it, would get, frustrated and so on. So we decided not to grade them on the quizzes that are embedded in Labster. So we graded for completion. So if students completed the whole simulation, then they will get a grade for it, just to see how they did. And I'm happy to report they did very well. So the lowest score would be 70-something% out of 100. So, and most students did above 80. And this was not paying that much attention because they knew they were going to get the full credit anyway. So the next time we are going to actually grade their work in Labster, because it seems like they are doing well. So yes, indeed, in the dashboard you can see how many intents the students did and what was the percentage of correct answers and so on. So it's a good way to monitor the students.
Yeah, and you can personalize it, teach as well, like you say, to adjust it to what you want to, like if you want to grade on completion or just how well they did, how many questions they got right and so on. All right, I think we are moving on then to the last question here, which is what advice do you have for other professors? So you've already given a lot of advice, let's be honest, but if there's anything else that you want to say, I think that would be great.
I think, it's the first thing is to try it, and that's a nice thing about Labster, that you sign up and then you are able to demo the simulations yourself. So I think it's a good, and they have so many. So it's really, like a smorgasbord. You like Swedish table. So you have a lot of options, and I would encourage you to try them and see, how interesting they are. And again, flexibility. So another thing that we appreciated in Labster is the flexibility. So especially, I'm not going to say name, but certain like textbook companies, they would provide all kinds of online tools and resources, but it's very packaged, like this is the way to use it. And you're like, well, I know my class, I know my students, I want to do it my own way. So in the case of Labster, we said, well, let's try to grade for completion. Let's use this, this, and this one. And so it is flexibility that was really one of the things that was attractive to me, that I was able to decide how I wanted to apply Labster in my course.
Great. Yeah, great advice. And now I think, unless you have more advice, but maybe some will come up. We do have a couple of questions here that I want to get to that I think are really good. So one is an important part of the microbiology labs is to build teamwork skills. And is there less teamwork building using Labster?
Well, Labster is a very individual experience. That said, you can build teamwork. I mean, that is a great question, and actually something I haven't thought too much when thinking about the online lab part. But if you are going to go online, you can probably do group work, and I wonder if maybe we could add a few elective Labster labs. So that would be the required one and an elective, and then students could, share that experience in a group. But that's a great question, and indeed, Labster is individual, not in a group.
Yeah, and I just want to add here, because it is something that we do get asked a lot actually, is, what about collaboration? What happens when you start looking more into a screen? And what we hear from professors, that there's a big difference, first of all, depending on how many students you have in the classroom and if it's physically in the classroom or if it's online. But we do have teachers using it, where several students sit in front of the computer and do it together actually, which is one way that actually causes more collaboration. And another thing, I had a professor with 2,000 students online, and you think, okay, that's pretty difficult to get people to collaborate like that. But what they do there is then take, make little groups where they use the lab as, preparing basically for them going in and then working as a group to answer questions that are more sort of, critical thinking or higher order thinking questions. So there are lots of different ways to do it, and us at Labster, we love to hear what you guys out there are doing, because it is an inspiration to everyone else. Because with something like this, it's relatively new with technology. It's great to hear new use cases all the time. So keep those stories coming our way, and we'll be sure to share them in webinars like this, so you can all learn from those ideas. So another question that came in here is, have you been able to quantitate level of competency via exam grades, etc., from students comparing total virtual versus total hands-on?
As I said, we used a quiz addressing a specific, lab activities that were done both hands-on and with Labster. We were thinking of redesigning that quiz and addressing a few more deeper concepts to see, besides the specific question related to that particular activity, but also if they had a deeper understanding. For hands-on, I don't know. We haven't really thought about it much because, again, the final goal for us, or the initial goal and the final goal would be to use it as a fully online lab. So that would be a bit hard to, to quantify.
Mm-hmm. Yeah. All right. You still have a few minutes there to type in your questions if you have any. I'm just quickly going to share some practical information because we did get a question here about the simulation, the microbiology simulation, the new ones, the microbiology simulations. So, like I said before, I'm not going to sort of go in and talk about all the new simulations because there are quite a few, but they do launch on September 30th, so that's next Monday. They'll be fully available for everyone to use, and then you can get access to the screencast like the one I showed you before, and all the quiz questions and so on the faculty resources page. And yeah, what else is there to say? Yeah, and there's a handout in this toolbar as well, and I'll send that out tomorrow with email as well in case anyone wants to read more about those simulations. So it looks like we don't have any more questions. I'm really happy that the ones that we did get so far were really great, I think, and thank you for sending those in. Let's just, yeah, that's it. Thank you.
I just remember one thing, Maria, sorry.
Yeah, go ahead.
If those who teach online or have an online shell, there is a way to insert Labster in your online shell, so that makes things even easier, so you don't have to always go in back and forth. So I just remember that now that we were talking about the practicalities, so that makes things easier.
Yeah. Yeah, we do have all kinds of integrations and access. The easiest way really to find out how to get to see all these things as well is to just reach out to us. You can also reach out to me, Maria at labster.com, if you have any questions and how to. Oh, let me see. I got one more question here. So I got one here. I think the attire of people in the exercises needs to be more compliant to safety rules. In the current image, the woman wears slippers and the trousers are not at full length. That's a great point as well. This is actually not a lab. This is a, I guess, like a clinic or something, but it's a part of the sort of the introductory scene of the simulation. But it's a great point. And again, I'm not the microbiology expert. I don't actually know what the safety rules are, but it's a great point. And yeah, and I think that was all for the questions then. Do you have any last remarks, Anna, or are you any last?
No, I think my contact info is somewhere in the beginning. So if you have any specific questions, as A Barral, ABARRAL at anu.edu, feel free to reach out if you have a specific microbiology question.
Yeah, that's something you're way better at answering. I do want to say we do have a bunch of microbiology experts and scientists at Labster as well. So I'm just representing here and not so much being the expert. But I hope you can bear with me, and thank you for that info. I'll send that your email along with the email I send out tomorrow, so people can get it there if they want. Everyone's going to receive that email, so yeah, look out for that in your inbox tomorrow. And without further ado, I just want to say, thank you so much to you, Anna, for taking the time to come in and share your Labster experience with us. We really appreciate it. And I'm sure everyone here in the webinar, I got a bunch of thank you messages here in the question section. So so thank you for the webinar, and it says it was very helpful and interesting. So thank you for those comments, guys. Thank you to everyone out there for joining. I hope you do want to join again for another webinar in the future. You can sign up to our newsletter, labster.com/newsletter, to get notifications and invitations to those. So yeah, thank you again to everyone.
Thank you, Maria. Thank you,
Thank you.
everyone.
Have a good
Bye-bye.
day. Bye.